അതുകൊണ്ട് ഈ സാധനം മാറ്റല്ലേ ഇവിടുന്ന്. ഇത് മാറ്റുകയേ വേണ്ട. ഫ്ലിപ്പ് ചെയ്യണം. സ്റ്റാർട്ട് അവിടെ ബാലികടയിലുണ്ട്. വെൽക്കം ബാക്ക് എവരിവൺ. അതൊന്ന് പറയാം. ഓക്കെ, അത് കഴിഞ്ഞിട്ട് കോൺഫറൻസ് കോൾ ഉണ്ട്. ഫസ്റ്റ് ഓഫ് അത് പ്രസൻറ്റേഷൻ ഒക്കെ അവിടെ നിന്ന് ഇത് വന്നത് ക്ലാരിറ്റി വന്നത്. പക്ഷേ ഇതെല്ലാം കറക്റ്റ് എടുക്കും. നിങ്ങൾ മാറി മോളെ ഓക്കെ. അവളുടെ ഫേസ് ഒന്ന് നോക്കണം. കറക്റ്റ് കോൺഫറൻസ് ഹാളിൻ്റെ ഫസ്റ്റ് ഫ്ലോർ. ഇപ്പം എങ്ങനെ വരുന്നത്? സമയം
മറ്റേ പാരലൽ സെഷൻ സ്റ്റാർട്ട് ചെയ്യണം.
നിങ്ങളുടെ കയ്യിൽ 2 ബുക്ക് എന്തെങ്കിലും ഉണ്ടോ പിള്ളേരെ?
Welcome back everyone. Now let's proceed with the first technical session of the day. So, simultaneously, the participants were restored for the second technical session by വേറെ ഉണ്ട്. ശരി. Please move to the conference hall of the first. കോൺഫറൻസ് അതിൻ്റെ ഫസ്റ്റ് പബ്ലിഷ് ചോദിക്കുന്നു.
ഇത് ഇച്ചിരി
പ്രസന്റേഷൻ ചെയ്യുന്നത് മാത്രം പോകത്തില്ല, എല്ലാവരും പോകത്തില്ല. താങ്ക് യു. മറ്റേ സാറിന് പറഞ്ഞാൽ സാറിന് എഴുതി വെച്ചിട്ടുണ്ടല്ലോ. ഇല്ല, ഞാനപ്പോൾ ഉണ്ടാക്കി. അത് ഓക്കെ ഓക്കെ, അതുപോലെ ചെയ്താൽ മതി. AC നമ്പർ
സ്റ്റാർട്ടായോ? ഇത് ഓൺലൈൻ
ലൈവ് ഇപ്പം ഇങ്ങനെ അവിടെ വെച്ചാൽ ഒത്തിരി അടുത്തായി.
അല്ലെങ്കിൽ ഒരു കുട്ടി അവിടെ നിർത്തണം, പഠിച്ചുകൊണ്ട് നിർത്തണം. അത് മീൻസ് അത് വേറെ സ്പീക്കറിൻ്റെ മാതിരി കാണാം. ആരെയും പിടിച്ചോണ്ടിരിക്കണം. അതാ
പറഞ്ഞോട്ടെ, അല്ലെങ്കിൽ ഈ സെഷൻ തുടങ്ങും.
സെഷൻ സ്റ്റാർട്ടാണ്.
സൈമൾട്ടേനിയസ് പറയണ്ടേ?
സാമൂഹ്യം പറഞ്ഞു
ഇതങ്ങ് പറയുമോ? ഇനി എല്ലാവരും മിണ്ടായി. ഞാൻ പറഞ്ഞു
അവിടെ ഓക്കെയാണ്. അവിടെയാണ് സെലക്ഷൻ ഉണ്ടാവും.
ഇപ്പൊ പൊട്ടിക്കാൻ പോയിക്കവര്. അപ്പൊ ഈ സെഷൻ നടത്തുമ്പോൾ ഇടയ്ക്ക് പറഞ്ഞു ഈ സെഷൻ പറഞ്ഞു. അപ്പൊ ഈ സൈമൾട്ടേനിയസ് പറയണ്ടല്ലോ. അപ്പൊ സൈമൾട്ടേനിയസ്ലി പേപ്പർ പ്രസന്റേഷനെ കുറിച്ച് പറയണ്ടല്ലോ. പക്ഷെ അവിടെ ആരും ഇല്ല. ഫുഡ് കഴിച്ച് കഴിയും തോന്നുന്നു. ആഫ്റ്റർ 1 hour after half an hour presentation.
2:30 will start the parallel session.
Conference after half an hour.
Conference is over there.
2:30 പറഞ്ഞോടാ. half an hour conclusion by 2:30 p.m.
Conference is over there.
ഇത് പറഞ്ഞില്ല സാറിനുണ്ട്. സാറിന്റെ പ്രസന്റേഷൻ നടക്കുമ്പോൾ അതിന്റെ ഇടയ്ക്ക് ഇവർ ഇറങ്ങി പോകത്തില്ലേ? അപ്പൊ എനിക്ക് വൃത്തിയോടാകത്തില്ലേ? എല്ലാം ഈ സെഷൻ തുടങ്ങണം. നമുക്ക് ഇടയ്ക്ക് വിളിക്കാം, രജിസ്ട്രേഷൻ വിളിക്കാം. തുടങ്ങിക്കോ. എൻ്റെ പേര് ലഭിക്കുവാണ്. സൈമൾട്ടേനിയസ് അപ്പോൾ പേപ്പർ പറയുന്നത് തുടങ്ങിക്കോ. ഓക്കെ, ഏറ്റവും സ്റ്റാർട്ട്.
ഇത് മ്യൂട്ട് ചെയ്യണോ? നിങ്ങൾ സംസാരിക്കുന്നത് മ്യൂട്ട് ചെയ്യണം. ലൈവ് ഉണ്ട് ഇവിടെ. അല്ലെങ്കിൽ വിട്ടിയുടെ വല്ലതും പറയാമല്ലോ. അതാ പറയണേ.
ഞങ്ങളോട് ഡിസ്കസ് ചെയ്യുന്നതെല്ലാം കേൾക്കുന്നത്.
പിന്നെ
ഇത് വർക്ക് ചെയ്യുന്നില്ല. അത് പോയി, ഇത് പോയി, ചാർജ് തീർന്നു.
Welcome back, everyone. Now, let's proceed with the first technical session of the day by Dr. G. Balasubramanian. For that, may I invite Mariama Philip of First PG Food and Industrial Microbiology to speak a few words about the speaker, who will take charge of the section and lead us through the forthcoming presentation and discussion.
Dr. G. Balasubramani is a distinguished researcher in microbial biotechnology, currently serving as Assistant Professor at the Department of Biotechnology, Savida School of Engineering, CMATS, Chennai. His research focuses prominently on antimicrobial resistance, natural product-based drug discovery, marine bioresources, nanobiotechnology, bioremediation, cancer biology, and immunology. Over the years, he has held key research positions, including Senior Research Fellow at ICAR Siba, Chennai, and Postdoctoral Fellow at Tianjin University, China. Earlier in his career, he contributed as a University Research Fellow and UGC Project Fellow at Peria University, Salem. He completed his PhD in biotechnology from Peria University, where his work introduced a novel approach in evaluating the antibiotic potential of fish-associated bacterial metabolites. His academic qualifications also include an MPhil, MSc, and BSc in biotechnology, supported by hands-on medical laboratory training. With extensive experience in marine-based therapeutic discovery and eco-friendly nanotechnology, Dr. Balasubramani continues to drive impactful research with high translational value. His contributions reflect both innovation and dedication towards societal and scientific advancement. We are honored to have him here with us today.
Thank you, Mariama, for praising about the speaker. The participants who are registered for paper presentation, please move to the conference hall at the first hall. The participants who have registered for the paper presentation, please move to the conference hall at the first floor. Thank you.
Good afternoon to everyone. I think it is difficult to make you head up with my presentation after the meal. So try to bear the. So I showing something for entertainment, not for infotainment. We will have a good interaction. So now how I reached it, can you see some fun over here? I think the first row noticed it. How I reached here, see, by bus, second series, so I reached here. Okay, see, here, what's my topic here? Curbing the arrogance of multi-drug resistant strains, immunocompromised host by quorum quenching bacterial police. So, as we are a human, if we are a stealer or robbers, who will control us? Policeman. Likely, for bacteria also, if there is a virulence or any pathogenic factor, then the other bacteria can control our. How? By secreting some enzymes like what they have. Stick, lock, gun, all and all. In the same way, the bacteria will work on another bacteria to control their virulence or pathogenic traits like biofilm formation, all and all. So that is said to be quorum quenching mechanism. First is quorum sensing, then quorum quenching. Quorum quenching is to arrest all the virulence factor. Quorum sensing is to how the bacteria getting resistance over antibiotics. Got it? So, see, simple. Just police and the stealer. So I will start. How we are going to curb this resistance? We have many mechanisms to control. As you all know that antibiotics not going to work in future. Though it may work, we may definitely face some consequences, right? So we need to go for an alternative. In what way we will find an alternative? Obviously the natural source. So all may stick on what? Plant derived one, or we can't sacrifice more natural things. So obviously we have to go again with the bacteria only. It's not a ending task. So it's always a recycling one. So we need to find a good thing. How? By having a eco-friendly nature. So how we can make eco-friendly nature? We have to keep our environment very clean and very clean. That is the only case. We can curb all these restrictions. As Sir told now in the morning, everywhere will be restriction. Don't touch here or there or like. See, in our days, in 90s, we used to eat soil. When we are childhood, when we play with the soil, when we eat soil, it won't hurt anything anymore. We won't go hospital. Someone asked whether it's good to go hospital, waiting at large. We have many pet clinic. There are, without any patients, they are sitting simply. You can go and refer them. There is no cost. Even he is also very, healthy. He may have not faced many problematic patients. Always we are going to big hospital for big cases. That is also my answer for the question. So go. Small problem is go. You don't need to wait. Anyway, you have to admit. Only that case you can go for big hospital. So there are pet hospitals are there to go. Okay. So now who is immunocompromised? Someone who underwent some surgical history, or they are very prone to get antimicrobial resistance, even though when we administer a specific type of antibiotics. So what are antimicrobials? When as organisms evolves for sometimes and change, if there is a change, then it may show some irresponsibilities to the antibiotic. What is the irresponsibility? How we are behaving in front of teacher inside classroom and outside classroom. The same way. Okay. So it is beyond. You must think which bacteria will be more impactful, which may not be. We can treat the bacteria with the infectious bacteria along with infectious bacteria, but sometimes it may come to normal. We cannot predict it. That's why bacteria becomes a recycling unended task. We always be aware with this task. So simply I am going with the immunocompromised host. Who are immunocompromised? See that cartoon. I am afraid neither your insurance nor your immune system will cover it. What it mean? Whether you will alive or not, whether you can claim insurance or not, if you are having some infection and you met with antibiotic resistant death. That's why this dialogue. Apart from underwent operation, if you met with, if you didn't consult doctor, obviously if there is a death, who will found that reason behind it? So you cannot claim that it's a natural death. Who will give for natural death? For insurance and all. See, so you have to be very keen when you go hospital. So see, the antibiotics, when it started in 1920, they introduced antibiotics. After that, we are very smooth. When times evolves, when we become hygienic with our environment, then all infections start to cause. See, most cases we are becoming hygienic just with the sanitizer. After COVID also, before COVID also, some people are very hygienic, don't go there. So we must immunize. How? We have to expose to the situation. Even nowadays, we are not going to the sunlight. Have any people continuously 3 days or 5 days, have you went under sunlight before 10 o'clock? You have been caught and seated under this room. Then how you will get immunity, all and all? Then you are saying that you should not touch anywhere, you have to be hygienic, all and all. You must immunize. How? You have to fight, you have to allow your body flora to fight against the external flora. Right? So, it's not in our hand. The nature has to change it. So here, see the rise of antibiotic resistance. When
over 1990 and 2020. Before we were healthy, you know, as I said, no, 1990s, those kids were very healthy. Nowadays, due to food styles, how one become immunocompromised, suddenly kidney problem, suddenly something else. Why? Only due to modern food styles, junk foods. All and all is a indirect cause for this immunocompromisation. Got it? So be healthy, have good food. So now I am going to see rise of antibiotic resistance. So how it impacts India and all around the world. So see, the multi-drug resistance causes every 15 seconds someone dies from an antibiotic resistant infection. Then see, in annually 7 lakhs MDR, 700K followers, like 7 lakhs people been died by antibiotic resistance infection. Then in 2050 there is a study estimated that there will be a case of 1000x resistance factor will be developed by the AMR strains. 1000x resistance factor. See, that may surpass the cancer. If there is a cancer death is 50,000, then in 2050 how much? 10 lakh 50,000 or crore times bigger. You just imagine the factor, resistance factor. So immunocompromised patients are include who are all immunocompromised. 65% burn victims and transplant recipients. Those are the immunocompromised host faces severe risk of MDR. SARS said no associated antibacterial deaths and attributable antibacterial deaths. These and all witnessed recently in Chandigarh. You see, these are the data we obtained from a survey conducted from 1922 and 2021. Here in 19 as in the morning sir mentioned there was associated deaths with 4.95 million deaths, whereas attributable, it is the sole cause for the multi-drug resistance. This one is those who underwent surgical history. So the death was quite high in immunocompromised host. That's why we have to be alarmed. When you are underwent any surgery, you must be with the uttermost care. See the death cause. Then who are the responsible for these things? Who are the responsible persons for these things? As we know, MDR strains, how it getting that MDR multi-drug resistance? There are certain bacteria remains persist, that is remains or persist. So those are said to be bacterial persisters. Even after treatment with antibiotics, some may grow or some may slowly grow, some may not grow. So it may undergo such stress. Those bacteria undergo such stress. So even after, if you remove the stress also, if you face, see that fourth point, the stress, they are growing or slow growing and can contribute to survive under stress conditions such as antibiotics, reactive oxygen, acid, starvation, even temperature or any carbon source or like supplements. So after stress removal also they can continue to grow and remain sensitive to same stress. What does it mean? What does it mean? It is always somehow potential to come back, right? Slowly. So that will communicate to the neighboring species that they may make us so film than earlier. So those are said to be biofilms. What is biofilms? You know, right? There is a matrix produced by the bacteria that will colonize and prevent that attack from antibiotics, and they survive for a longer period. So we have to attack biofilms producing bacteria. Those are the major threats for this MDR. So how we can curb? Through quenching. The infection causing mechanism is through quorum sensing. See, this and all I will skip. So recently you can see August 21-25 there was a data from Chandigarh. There was critical threatens, superbugs threatens hospitals as antibiotic resistance rises. You know, there is a scientist who named Shafiq. He conducted a survey named antimicrobial prescription patterns in tertiary care centers in India. A multicentric point prevalence survey. See, as by the Society of Antimicrobial Stewardship Practice in India. See this one. So why they have conducted this one? Whenever we go for hospital, even for a small cut, whatever the prescription they suggest is not working. That leads to a overdose of antibiotics. Then that is the cause for MDR. As we saw, we have witnessed many things. Even for simple headache, if you go medical shop, they will give 5 tablets. If we ask this for that, that for that, this for this, that is our overdose only. We need a required thing for a required purpose. Simply just to sell. As Sir said, no, only 3 days. For 5 days, if we buy only 1 day, we'll. Obviously, the 4 remains wasted. So these are marketing strategies from pharmaceuticals things. So why they conducted this study? Many people have faced antibiotic resistance over different hospitals. You know, they mainly focused on AIMS. 8 AIMS different provinces. So who from Jimmer, you know Jimmer. He is a leading person who conducted that survey, and he conducted those studies over 3974 patients in 8 hospitals. All he focused with AIMS because there only we will get good and hi-fi and sophisticated treatment strategy protocols, right? So he surveyed over there. Mainly they found that the ICU, intensive care unit peoples are prone to get these failures in antibiotics treatment. So see, out of 10 people in the world getting common antibiotics such as ceftriaxone and metronidazole in ICU. Over half period, most of the drugs been watch groups. There are categorized into 4 types. Watch group, see that bolded thing. Watch group, those are said to be severely treated with utmost care because it was, as I said, no last resort antibiotics. So we must use them with utmost care because it may speed up the resistance factor, the one which you are going to apply. So some groups been categorized as watch group. Then others reserve drugs. Reserve drugs also causes the same, but there won't be any alarm. But there are risk for a resistance factor. Then other thing, empiric. You know empiric what it is? What is empiric? Sir said no empirically. In certain cause of diseases, we may not know which drug to be administered. Only with some group of drug, they will simply administer without any lab test. For the quick action, they want something to be, resolved. So at that time, they will empirically suggest prescribe some tablet, some drugs. Then irrational drugs. Something may be too much, something may for too long. Too much drug and too long. That also leads to antimicrobial resistance. So what is that? Irrational. Then redundant combos. We may not require such, as I said, in medical they will give some tablets like that is redundant compounds. So overall, after administering these sort of drugs, they found that culprit as E. coli and Acinetobacter. Over there from that 8 institute, major cause for E. coli and Acinetobacter. So why they want to do that survey? In Jimmer they used to always suggest a strong dose. As I said, no, the resort drugs always they will end up with the results. If they fail, yes, then they want to find a reason behind it. So that's why they underwent this survey in Jimmer. So, India's specific concern. See, due to this antimicrobial resistance, around 30 newborns in India die annually in ICU because they are resistant to drugs. Then, high antibiotic usage. I said no, watch group drugs. Now I listed something empiric, irrational, redundant, all and all. So that is the high antibiotic usage. Lack of infrastructure. Inadequate healthcare infrastructure such as lack of diagnostic tools leads to doctor prescribing broad spectrum antibiotics empirically contributing to resistance. Then infection control. Overcrowded and high hygienic hospital with poor infection control promotes the spread of resistance bacteria, making hospital acquired infection a significant problem. So I will give one example story. When I was in China, we were under control of COVID, right? So I have to come to India. So see my mental states, I have to reach, the only thing I have to reach India. But what they suggested, when I step down, don't touch that pole, iron pole. When I use inside lift, don't click. Then how should I, when I go to the shopping, I have to pay for 2 days to get flight. Suddenly we can't get a flight. So one day I must be there. So wherever I go for shopping, then they always used to wear gloves because their conditions will be chill. So even then with this chill condition, whatever they touches, it's contagious because someone may touch like this. So if they take fruits also, it will be contagious. So we cannot, whenever I see the vegetable, that also I looks like a virus because if I eat suddenly I will go away. So I must be with utmost care. So see the thing. So we were in utmost care. Then during that transit, I met around 3 medical people from Tianjin. After 12th, they go for medical studies. Over there, I convinced them that we will go India, then we will inform them we came from, then they will seclude us like. They agreed. The 3 people, I only made a canvas with, then we went to the Trichy airport. The same podium like this in Trichy. The people who enters from airport, from flight station to enter. They simply standing here. That time we can detect only through thermometer. They had only 2 thermometers. We were standing, sir, we came, we want to check all and all. Simply people, others are going because they were not aware about those infections. We only voluntarily addressed that, then they are searching for the thermometer. They found one down, then they are detecting it. When they detect, they found it's no charge. See how this infection spread to our COVID, how many people lost parents, all and all. Simply, then after they went to the immigration, they are calling from, there will be a down, accelerator or something here. They are calling the names from upstairs and down. Meanwhile, they might have went to hotels, all and all they drink with the sipping, all and all. See how contagious the COVID, how that, see simply with the lethargicness, we lose many things. So contagious is not a simple thing. We faced with the virus, we didn't face much with the bacteria, but silently, SARS said no, silent pandemic, it's happening. We cannot ensure that, we cannot identify where and when. Okay, so I don't know where it will end, but I will quit session soon. Then here we will see about quorum sensing. What is quorum sensing? Before I said how these virulence or antimicrobial resistance being produced. Now quorum sensing. So see, quorum sensing is a cell-to-cell communication among bacteria that regulates gene expression by producing small extracellular signaling molecules known as autoinducers. So we know bacteria is there. We have 2 classes of bacteria, gram positive and gram negative. So each may produces different autoinducers. We will see what it does. When it is low cell density, there won't be much impact by the bacteria. When it is grouped, as I said, biofilm, when it forms a community with a signaling molecule, they are very violent against any external agent like antibiotics, enzymes, all and all. So that is how the resistance mechanism being developed. So how we are going to see the polymeric matrices that resist antibiotic diffusion, antibiotic interaction with the polymeric matrix that reduces antibiotic activity, enzyme mediated resistance such as beta-lactamase, alteration of metabolic activity within the biofilm, the effects of antibiotics by efflux pumps. Then genetic modification of target cells or hiding target sites. This is how it will work against antibiotics, the biofilms. So see the signaling molecules from gram positive and gram negative. Here the signaling molecules are said to be in gram positive auto-inducing peptides, whereas in gram negative the signaling molecules is AHL, N-acyl homocysteine lactose. So both, when some external agent comes, both may sometimes communicate each other by auto-inducers too. It was a big task, just I am skipping, limiting that content. So there is a pathway how that gram positive becomes virulent. So some gene must be expressed. So each bacterium has its own. Here here we can example a gram negative Vibrio fishery is the first one we discovered for this quorum sensing. In that they found LuxI, Lux R genes are responsible for the expression, gene expression. Once the gene been expressed, then they won't allow any antibiotic to attack in gram positive, whereas in gram negative. Whereas in gram positive, it is sort of oligopeptides. So once that oligopeptides, once the cell been matured, then it will pass through a cell membrane via ABC transport membrane. Once that AIB threshold level reached, it will activate the kinase and histidine kinase sensor kinase. Then that histidine sensor activates kinase. Then these thing phosphorylates again with histidine residues. Then the gene expression activates. It's quite tedious and tough with each individual bacteria. So we must study whichever the bacteria we target. That is the only thing underlies in quorum sensing and quorum quenching. So here how these quorum quenching bacteria fights against quorum sensing. So through three major categories. One is AHL lactonase, AHL acylase and AHL oxidase and reductase. See what it's doing here, which hydrolyzes lactones. Then AHL acylase, which hydrolyzes the amino groups. So once it's get targeted, then we may ensure that virulence factor or biofilm or motility being suppressed. So this is how the action will be done by the quorum quenching bacteria. See, this is gram negative and gram positive. Some pathways like a gun point we have to shoot, where we have to attack over some expression point when it activates certain things. The main thing to target is auto-inducer in gram negative, whereas auto-inducing peptides in gram positive. These two are our targets. When we stops from the beginning, then there won't be any expression, there won't be any virulence, or there won't be any biofilm formation. I don't want to go much. See, only thing we have to target biofilms. In what way? We know some bacteria will work. As I said, some may be opportunistic, some may be potential to fight against another potential one. But how we can do even better? So if we synthesize a nanoparticles, because it was an untouched study, I only conducted for the first time. If you refer G. Balasubramani quorum quenching nanoparticles, mediated nanoparticles, the bacteria must be screened first for nanoparticle synthesis. Then that nanoparticles works against. I am the one first initiated in this. There were bacteria for nanoparticle synthesis, but if you try at large for your future projects and all, then definitely it will look, or else we will come out with a good findings. So I conclude that that policeman must wear a brown shirt. Brown shirt here, Kerala. So likely the golden, any nanoparticles must be encapsulated. So that is my conclusion. Who is the what is the thing here? If I and evidence that something is happening with the quorum quenching mediated nanoparticles will be the source. So you may also try, if you have good guide, or if you want to approach me for your future studies, then you can come. So that's it. I don't want to stretch a lot. So these are some of the common entrance exam you can avail for your reference. That's it. The only thing I want to suggest to fight against biofilm producing bacteria for the MDR is quorum quenching bacteria. Once you found quorum quenching bacteria that produces oxidative reductase, acylase and all enzymes I have shown, lactonase. So if you screen that bacteria, that would be the source to synthesize nanoparticles. Now everywhere nanoparticles synthesis is a project, but we can introduce with a new source. However, before a big task through quorum quenching screening, that is a quite big task. We must ensure whether it's quorum quenching or not. That is the main task. So if you found such bacteria, then we can use as a source. So that will act as a policeman to control these MDR strains arrogance. That's it. Thank you.
This section is open for discussion.
If you have doubts, please.
just have, first of all, thank you for this wonderful presentation. I'm of a different field, so I just wanted to know, like, when we are giving this antibiotics, this bacteria will get this resistance. So earlier speaker also mentioned that you should use this conventional things, the ginger and all those things. So how come the bacteria is not adapting to that conventional thing when compared to this?
That's why we have to be, always you are starved to find something. So it's not an ending case. As I mentioned, no, we must stick to some other source, whichever works well. Then we will stick for a while. Then once it gets that resistance, then again we have to go for. There is no option to end up with.
But I think with the conventional treatment techniques, it was like working for...
No, due to the conventional treatment, due to our usage of, because some people prescribing though intentionally, but may not know what to be suggested. Which infection type, as I said, no, it must be screened before which infection type it was. Then only we must suggest, as we go, simply ask, she is 18 years old. Even for small fever, cold and all, we are getting some prescription from medical itself. That must be avoided, first thing. Then when you go hospital also, you must consult a senior consultant. It's not simply whoever is training, you can go. But you can, if you go first time, if it doesn't work, then obviously you will change, right? That won't be the next solution. The medical, prescription suggested by the people matters a lot. They should know what to be done. Unnecessarily, if something they suggest this, it leads to some drastic.
I'm just asking about the phytochemicals and
That from that you
is not able to adapt accordingly. Like
no, definitely something will be adapted. But thing is that already there will be marketized products. There are some products to be administered. We cannot rise up with suddenly, as said, no, empirically, Covishield and Covid, something they gave without any pre-treatment, this because it was pandemic, so they must treat something with immediate solution. Likely something will give some solution. Until then we have to screen, otherwise research will stop. There won't be any research. If we are under control, then we don't need to do research. We need to study, something will happen in nature. So that's the thing. No, see, always we cannot rely on natural source. Obviously, whatever we find synthetically, we have to do. The synthetic origin may not change at any cost, whereas the natural origin may have changed when there is a heat. Some heat also may have some changes in that, even pH, temperature in that. But synthetic, once it be designed with this many pH, it will never ever change. So whatever the result, it will show the same result. So synthetic ones are always a problematic one. See, the consumption is quite vast. I simply quitted because as you felt boring, all are yawnings, like gulping me like. Only some people, see, even then now, see that. That's why I quitted soon. Thank you so much. If you have any other questions, please.
Sir, I will add on to what he has asked. Maybe phytochemicals, it is a mixture of compounds. The antibiotics what we give is a single compound in large doses. So since it is a mixture of compounds, the bacteria may be little bit confused. Getting confused means different class of compounds are there with multiple modes of action. So maybe that is why. And also natural compounds we are not using in very high concentrations as in the case of antibiotics. So maybe that is one reason for not developing resistance against the natural compounds. Thank you. And sir, you were talking about quorum quenching bacteria. Are they effective against all kinds of India, or is it specific to certain species?
Certain species, we have to target some pathways, ma'am. If you want to focus on pseudomonas, because we must have some thirst area, which infection is prone at large. So if you want to focus pseudomonas, so I have an example over here. See, we have to target. See, we have to target the less IOR RH gene. If we target that if you gunpoint over there, then the expression won't be there. So obviously, yeah, species specific and pathway specific. So we have to target only the pathways. It's not dealing one antibiotic, 5 different bacteria. I done antibiotic sensitivity test, so it will reveal some result like. It must be because some, Klebsiella, Sar morning said no, pneumonia, Klebsiella pneumonia, we can work over there. If you find a pathway, then it is good, easy. Even then, it will adapt the one which we are going to target with. That is the quorum sensing inhibitor. That inhibitor also may be sensitive to that quorum sensing signal molecules. The signal molecule, when days goes, when the emergence evolves by the pathogen, when days goes, you keep in mind, evolution, that may change that target specific expression also. That also a case to arise. But we must read all those. It's not never-ending task. One thing, there may be a solution, but pharmaceutical tactics are there. Whatever they produce, they can't simply throw in the trash or like something they must administer. Thank you, sir.
Thank you, sir, for an insightful presentation on innovative strategies to combat multi-drug resistant infection through the disruption of bacterial communication. Your discussion on the evolution of antimicrobial resistance, the role of bacterial persistor, and the impact of high antibiotic consumption, particularly in India, was highly enlightening. I especially appreciated your explanation on quorum sensing in gram-negative and gram-positive bacteria and the promising role of quorum-quenching enzymes, including the synergistic use with antibiotics as a novel approach to reduce pathogenicity and combat resistance. Thank you, sir. Now I invite beloved HOD, Dr. Ambuli, to honor the Dr. G. Balasubramanian. Thank you, ma'am. It is my privilege to introduce our esteemed resource person, Dr. Tijo Cherian, Assistant Professor, MSc Food and Industry Microbiology, BK College, Amalagiri. Dr. Tijo Cherian is a dedicated educator and researcher in the field of food and industrial microbiology. His valuable contributions to academic development and student research have been highly commendable. With strong expertise and active involvement in the field, he continues to inspire budding microbiologists to explore innovative ideas and solutions in food science and industry. We are honored to have him here with us today to share his knowledge and insights through this session. On behalf of the organizing committee as well as the participants, I am delighted to welcome you, sir, and we eagerly look forward to your enriching presentation. An announcement is there. Those if anybody is in hurry or in emergency to go back, if there is any hurry, you can go back. You can, and there is a need to present the paper. Please go back to the conference hall for presenting it. Or otherwise, you can pertain over here.
So good evening all. I, Dr. Teja Cherian, serving as Assistant Professor in the PG Department of Food and Industry Microbiology, BK College, now will deliver a short talk on role of community awareness and behavioral changes in combating AMR. So from the morning till now, we are all just hearing about AMR. What it actually means. That is the antimicrobial resistance. So my thematic aim of the talk will be to learn about the awareness and the behavioral change that will be start from the community level. Since humans, we all are humans, we are living in a society. We are living in a close-knitted community. So how does AMR or the infections or the diseases affect on the total community as a whole? When we talk about a community, we talk about the advanced type of intimate relationships between the human beings. Introduction to AMR, I will not just go into the detail because from the morning till now, all are, I think, must have got a short idea that what does AMR means. Antimicrobial resistance. Overuse, misuse, or the over type of reliance on the medical type of therapeutic drugs or any type of medicine or any type of antibiotics. So the all around overall dependence on that will ultimately cause the antimicrobial species. Okay. In humans also we know our skin, our enterics. If you know the enterics means our gut microflora. This all are inhabited by the various populations of especially the bacteria. Okay. When you got any type of gut infection, so means what? Your gut is infected by various types of different microorganisms. Naturally, bioflora will be there. Means natural strains of bacteria will be there, but that will be not harmful for your body. Okay. So when the harm comes means if there are certain types of different type of strains that will cause the disease. Not all bacteria or not all the microorganisms, for example bacteria, fungi, protozoa, they are not infectious. Only some of the strains are infectious. The bacteria present in curd, that is lactobacillus, that is what? That is useful because when you consume curd or yogurt, so that lactobacillus will act as a probiotic source. Probiotic means that will enhance your overall digestion process. So introduction to AMR, just a small nap on that. AMR is the ability of microorganisms to resist the antimicrobial drugs. Causes: overuse, misuse, overinfection, prevention, and environmental spread. Overall, AMR overall threatens the global health, food security, and socio-economic development. Community action specifically and significantly influence the antibiotic use patterns. Now, why we need to have a community awareness? Why we all need to know about the over-reliance or the over type of dependence on the consumption of diseases. If we even simply get the simple cold or runny nose, what we will take? We are not going to take any type of natural sources. We all depends upon the medicine. That is the first line of dependence on the medicines or the chemical drugs. Most antibiotic use happens in the households and the outpatient settings, where decisions are often depend upon the knowledge and the beliefs rather than the medical advice. So we have medicine at our home. What we will do? We are first doing the self-medication. Sondamite namal doctor aguva. Okay, that is a first line of blunder that we are creating moving towards the AMR section. Why we want to use the doctor? We are not certified doctors. Nobody is a certified doctor here, unless you have the medical degrees. So the first line of defense is, if you are having a simple cold or fever, you should first go for the natural sources, not just relying on the medical drugs. Key misconceptions or communities. Antibiotics cure viral diseases like cold, flu, and dengue. This is highly blunder type of things that we are doing. Viral diseases can never be treated with the antibiotics. Antibiotics specifically target only the bacteria, not your virus. Consuming antibiotics during the viral disease is not going to serve any purpose. Fungal disease, they cannot be treated with antibiotics. Viral disease cannot be treated with antibiotics. Protozoan type of disease cannot be treated with antibiotics. Only the disease caused by bacteria can only be treated with antibiotics. No other diseases can be treated by antibiotics. Stop antibiotic once symptoms improve. When doctor prescribe you to do the course of 2 weeks, after 1 week your body is feeling comfort, so you will stop the medication. That is highly wrong. You should continue the time course that doctor has provided you. For 1 week, 2 weeks, 3 weeks, whatever course he has done, you should complete the course. Stopping in between the course will again reoccurrence of the disease will come. Okay, so prescribed time is 10 days. For example, if it is 10 days, so continue the disease for 10 days. On 11th day, stop the medication. No need to continue for more than any type of prescribed time. Leftover medicines can be used later. It's highly unsafe. Leftover medicines are not used for your after use. No. Whenever expiry date medicines are there, please don't consume that. Instead of gaining any type of advantage, your body will be at loss. All infections require antibiotics. Highly incorrect. Antibiotics, as I already told, target only the bacterial type of infections. Other type of non-bacterial infections can never be solved by the antibiotics. Misbelief continue to unnecessary consumption and resistance. Addressing these misconceptions are crucial for the promotion of correct antibiotic patterns. Drivers of irrational antibiotic use. So there are several social and behavioral factors that led to the irrational use or the rampant use of the antibiotics. First one is the overuse, misuse, poor infection prevention, and environmental spread. Overuse means you are overprescribing the medicine or you are overusing the medicine. Misuse means incomplete treatment. Incomplete treatment means instead of taking 1 gram sample of ampicillin, you are taking 2 or 3 or more. So that is the misuse. Limited access of healthcare also pushes the people towards self-medication. Social pressure and expectations from healthcare providers, lack of awareness, cultural beliefs and home remedies mix with partial scientific understanding, inadequate health literacy. Role of community engagement, what the community can do to raise the awareness against this. We can create the awareness through campaigns, workshops, local health programs. Schools, colleges, universities can act as a online or on-spot platform to do the conducting the drive or the awareness camp on the social as a whole, society as a whole. Involving school leaders, NGOs, veterinarians, pharmacists, etc. Encouraging community-based surveillance and reporting. Empowering individuals to make informed health decisions. So it means that community as a whole, one or all, has to participate or has to create a general type of moderate awareness on the AMR patterns. Behavioral change strategy. Our behavior should also change towards the outlook of the medical conditions. How to react to a particular or to a particular condition. Behavioral change requires understanding, can generally nudge the people towards the safer antibiotic behaviors. Health education, clearly culturally appropriate messages must be transmitted among the masses to overcome the patterns of AMR and to also hold the knowledge. Poster reminders at pharmacists or clinics or the wild sort of participation. Behavioral frameworks, COMP model, that is capability, opportunity, motivation leads to the changes in the behavioral patterns. Positive reinforcement, reward-based community programs, peer influence, testimonials, community champions. Promoting responsible antibiotic use. Complete the full course as prescribed. Don't share the antibiotics. One antibiotics cannot be taken by to the other. For example, one person is taking ampicillin. Other person should not take the same because one or two persons can have the drug allergy. As in my case, I have the drug allergy of clindamycin. I cannot take clindamycin. Okay, when I take clindamycin, my whole body will develop the rashes. So that transmitters means what? One drug can be suited to one, cannot suit to others. So every human body or every human psychology or human physique is a different physique. Don't share the antibiotics. We should never share one antibiotics to another. Avoid self-medication. That is the maximum persons here. We are all doing the self-medication. We are becoming our own doctors. Choose accredited pharmacies. Pharmacy also we should always go for the certified type of pharmacies or certified types of medical institutions for the purchasing and logging of the drugs. Good hygiene, timely vaccination and seeking guidance from qualified healthcare professionals also reduce the unnecessary antibiotic exposures. Community level infection treatment, have a proper hand hygiene, safe food handling, clear water and sanitation, responsible animal husbandry practices, waste disposal to reduce the environmental reservoirs of the resistant bacteria. The role of media and communication, combating strategy involves raising the full-fledged mass awareness program in order to deal with the problem of AMR. Promoting behavioral changes, that is to have a unique and a controlled site of how to use the drug prescribed to you. Improving practices, that is strengthening the infection prevention and the control. Advocating for the policies, support the development of AMS policies, that is antimicrobial sustainability policies. Initiatives, so till now, what around the globe, what are the initiatives has been taken? Global initiatives like WHO's World Antimicrobial Awareness Week, that is going on. Okay, so we have designed a seminar for 2 days, that is today and tomorrow, because 18 to 24th November is a Global AMR Awareness Week. Okay, It is coined and sustained by the World Health Organization. So around the globe, such type of AMR sessions, AMR conferences are going on around the globe. Campaigns should show how the public communication can change the behavior. Local community-driven programs and school-based education have also demonstrated the measurable improvements in the antimicrobial awareness. Challenges in community-based changes. What are the challenges coming here? Low health literacy. Kerala is the topmost literate state in the country of India, but our health literacy is very much poor. Misleading information spread via the social media. Resistance to changing long-standing habits, limited access to healthcare or health services, language, culture and socio-economical barriers. Opportunities, increasing the AMR's awareness into school curricula. We have to introduce a specific AMR awareness or a specific medical awareness in our school curricula. Empowering pharmacists as the community educators, use of digital health tools for education, partnership with NGOs and local government bodies, incentive-based programs for community participation. Recommendations, design evidence-based community awareness program in order to strengthen the overall MRR patterns in the prescribed wards and to also for the mass involvement or the people's participation in the drive against the AMR. Conclusion, community behavior is central in controlling the AMR. AMR controlling by me or you or only 2 or 3 persons will not serve the purpose. All community or all population has to come together for the critical role in deciding the action against the AMR things. Awareness plus responsible actions can lead to the reduction in the misuse of the antibiotic usage. Collective participation ensures sustainable AMR mitigation. Combating AMR starts with the home, with the informed communities. Thank you very much.
Thank you, sir, for insightful session on antimicrobial resistance. Your presentation highlighted the critical importance of community awareness, addressing common misconceptions that contribute to resistance. We appreciate your discussion on the role of community engagement through campaigns, workshops, and awareness programs, as well as practical behavior change strategies such as posters, reminders at pharmacies and clinics, and promoting responsible antibiotic use under certified medical guidance. Your emphasis on community-level infection prevention, the effective use of media and communication channels, and the initiatives taken by WHO to combat AMR provided a comprehensive understanding of the global and local efforts needed to tackle this challenge. Now the session is open to discussion.
Hello, teacher. If we go for allopathy medicine, what will be the consequences?
Pardon?
If we go for allopathy, what
yes.
will be the consequences?
No, you already we are using the allopathy now.
Allopathy. Homeopathy, sorry.
So this all is limited for the allopathic sections, okay? When homeopathy, there is only one rule. Either it will cause the benefit or it will not cause the benefit, but it will not have the side effects. Allopathy is not like that. It will cause the benefit, but long-term use will lead to the side effects. So AMR is one of the side effects of long-term use of the drugs. Also, the misuse or the overuse of the drugs. So homeopathy, it has a clear indication. Either it will do the benefit or it will not do the benefit, but it will not do the side effect. It has only one condition.
Thank you.
So homeopathic drugs are like synthetic compounds or natural compounds?
The homeopathy drugs, there are natural drugs also, plus the synthetic also. Both are there. Plus they can be mixtures also.
But the synthetic ones are said to cause all these things. So
Yes,
allopathy
yes.
can influence
Yes,
on
yes.
the
Allopathy is totally based upon the chemical structures. Allopathy-based medicine that we have always consumed, that all are composed of the synthetic based. Then nothing is true with the natural ones. Only homeopathy or the naturopathy or the Unani and Ayush like that, they only prescribe the plant-based used.
Sir, as part of the awareness programs, do you have the apprehension or do you share the apprehension that these awareness programs are often restricted to the scientific community and maybe even among the physicians?
Yes, ma'am.
But coming to the grassroots level, being the Antimicrobial Awareness Week, is there any program in Kerala addressing this issue or giving awareness programs to the common man?
Ma'am, I will take the example of today's seminar also. This seminar, we are the faculties, we are participating. Scientists participated, these students participated. Okay. So one type of, we need to have a grassroots type of level, that we have to start the AMR sessions or the medical, at least a medical education session from the childhood only, especially in the schools. Because school children, the psychology of school children is that they used to grasp any type of subject very easily, because it is their brain development stage, it is their cognitive development stage. So if we try to introduce such type of curriculum from our school days only, naturally when they will be turning into adults, they will have a cut and clear understanding how to use a drug pattern. So we need to start, or we need to introduce that type of thing from our school days only.
Maybe we can have as an outcome of this seminar, maybe we can give a representation to Karsap or something like that. This should be more
Sure, ma'am,
wide.
sure.
Thank you.
No, no issues, no issues, no issues. Cancer has a different type of cancer. I don't. AMR has no reference to cancers like that. But when you do the cancer, or when you go for the chemotherapy and radiotherapy, your immunity will decrease. When your immunity will decrease, ultimately the pathogens or the bacteria, any type of infection you can catch very easily. Okay, so AMR direct has no relation to AMR to the cancerous things. Madam, please.
The cancer patients will be in a condition to be take up more antibiotics in those third stage and fourth stage. So that would be the condition they are taking more antibiotics and prone to the resistant bacteria prevalence.
Plus that also depends upon the stage of cancer, plus the present health condition of the patient. Any other? Last, Sijo, no questions? Fahim, Afaf, you all are scholars. So thank you very much, thanks.
Now, those who have registered for oral presentation from the category of research scholar and faculty, please be ready with the presentation.
will present you first? You want to go first? Okay. Stage wants to go first.
Good afternoon all. I'm Fahim bin Zahid. I am from Yemen, research scholar in my first college. Today I will present my topic, exploring of the therapeutic potential of the ethanolic extract travelers stress and in vitro study, under guidance of Dr. Dina Merli. The travelers stress are also known as nirinil in Malayalam, also in Arabic language, al-hasak, nabat al-hasak. Indian folklore or traditional medicine, family Zygophilicia, the plant characterized by the flowers and spiky fruit that contain sharp spine. Used in traditional medicine system, particularly in Ayurveda and Arabic and Chinese medicine. The fruit is used as treatment for UTI and discomfort and pain. The phytochemistry of this plant, alkaloid, glycoside, saponin, flavonoid, and steroid. The objective of this study to extract the travelers straight fruit, qualitative and quantitative photochemical analysis in ATT to evaluate the antioxidant activity and cytotoxicity in vitro, to assess the antimicrobial activity against multi-drug resistant uropathogen, to determine the MIC and MBC value and SEM analysis, to investigate the antibiofilm effect of ATT, to identify major bioactive compound by use HRLCMS. The material and method, first one, extraction of travelers stress by succinate extraction, then use rotary evaporation and lyophilization. Qualitative photochemical analysis, I did the phenolic and alkaloid glycoside and flavonoid and extra. Also estimation the phenolic by Foley reagent method, and after that red absorbance by use the UV spectrophotometer. Then estimation of the flavonoid by aluminum chloride method. Then DBPH assay by use DBPH reagent, and FRAB assay by use the FRAB reagent. Then I did the ABTS. After that, did the MTT assay for measurement the cell toxicity of this plant by use two cell line, L929 and human kidney embryonic cell line 293. And did the MIC and MBC by microplate diffusion method. After that, do the biofilm assay by used Congo red agar. After that, did the antibiofilm by used test tube method. Then SEM analysis. After that, measurement the bioactive compound by used HRLCMS. The result, I got the following positive result for quantitative photochemical analysis like alkaloid, saponin, steroid, glycoside, flavonoid, and extra. The quantitative photochemical analysis test, phenolic content 7.9 and the flavonoid content 23.22. The antioxidant assay, compare my result with standard, the DBPH 46 microgram per ml, and the FRAB assay 30 microgram per ml, and the ABTS 41 microgram per ml. This result compared by standard. Cell toxicity assay with MTT assay for L929 and human kidney embryonic cell line. After 24 hours of incubation, the test compound not toxic against the both cell, L929 and human kidney embryonic cell line. The morphology of the cell confirmed this result. Determination of the antimicrobial activity against multi-drug resistant bacteria isolated from urinary tract infection. First one, I did the calculate the multiple antibiotic resistant MAR analysis of the bacteria by collect this by use this formula number of antibiotic to which the isolate showed resistant per total number of the antibiotic test multiple hundred. For example, the first one, Klebsiella pneumonia, the number of antibiotic resistant by bacteria 17, and the number of antibiotic which bacterial sensitive for, a total number of antibiotic 21, the MAR percent 18.95. And the another bacteria also, the high multi-drug resistant bacteria, Pseudomonas aeruginosa, the MAR percent 93.75. After that, I did the antimicrobial activity of the ATT by used well diffusion method was found to be against multi-drug resistant bacteria isolated from urinary tract infection disease, gram-positive and gram-negative. This plant effective against both the gram-positive and gram-negative bacteria. Gram-positive like Enterococcus faecalis and methicillin resistant Staphylococcus aureus, and the gram-negative like Klebsiella pneumonia, Escherichia coli, and Pseudomonas aeruginosa, and Acinetobacter baumannii. Then I did the MIC, micro diffusion assay to determine the MIC and the MBC compound against multi-drug resistant bacteria. Well, with the color change, this indicates the bacteria is grew, and the dark well indicated the plant is inhibition this bacteria. The MIC, after that I take from the first 4 wells and inculum to the molar Hinton agar and to confirm the MBC. This table shows the summary of the MIC and MBC. All bacteria, the MIC 100 except the Enterococcus faecalis, 200 milligram per ml MIC and MBC. The preparation of the Congo red agar for detection of biofilm forming bacteria and investigation the antibiofilm effect of ATP. This photo shows the black colony. The black colony in the Congo red agar indicated this bacteria able to do the biofilm in our body. And after that, used antibiofilm assay by used tube method. My plant, Travelus terrestris, inhibit the multi-drug resistant biofilm absorbed in the virus concentration of the extract. Then detection of inhibition present of the biofilm formation. This table shows statistical analysis shows the significance correlation will be value less than 0.05 between the extract concentration and the biofilm inhibition. The same result, the ATT caused major structural damage to the bacterial cell wall and cell membrane. The same confirm it is the strong antibacterial activity against both bacteria, methicillin resistant Staphylococcus aureus and Escherichia coli indicate affect the both gram-positive and gram-negative bacteria. The LCMS, I did the LCMS. This table shows the total ion chromatogram of ATT positive mode and this for negative mode. I get the total 117 bioactive compound was indicated 51 positive ion mode and 66 negative ion mode. This indicated the alkaloid and flavonoid and flavonoid and kistra. Many of this exhibit the antioxidant and antibacterial and anti-inflammatory and cytotoxic activities. This is the most common important bioactive compound after HRLCMS. This is the outcome. I got the this plant rich for alkaloid and saponin and flavonoid. Also, I got the good result in the antioxidant activity and no significant toxicity against L929 and human kidney embryonic cell line. And this test of ATT effective against multi-drug resistant bacteria and evaluate the ATT ability to disrupt the biofilm formation of the gram-negative and gram-positive bacteria. The same analysis confirmed the strong antibacterial activity against the both gram-positive and gram-negative bacteria. The HRLCMS analysis indicated the multiple bioactive compounds. Conclusion, the ATT contains multiple bioactive compounds constituted with the diversity therapeutic properties. This finding provides the scientific validation for the traditional use of Travelus terrestris, and the further study for this identify the compound might lead to discovery of the potential drug. The acknowledgement, I want acknowledgement for my research center, McFast College and Mahatma Gandhi University and Bushbagiri Medical College for assist with the sample collection and conference organizing. Thank you.
Do you have any queries? The way you organize the slide is very good. And the thing I have a doubt in MBC, which stain you used for color indication and next one. Last but first. Which is dye, stain, dye. Dye, stain. Resazurin. How you differentiate with different colors? What you try to say?
The dark color is my blank color. The dark color indicated that the dark color indicated the bacterial no activity, no metabolic activity. But the red color and the oral beige color, this indicated the bacterial has metabolic activity and live bacterial head.
Whereas in MIC and MBC, how you fix that 100 and 200?
My concentration starts from 200 milligram per ml. This concentration 200 milligram per ml. Second concentration 100 serial dilution. Serial dilution is 100 milligram per ml. This dilution called MIC, minimum inhibition concentration. After that, take from this 4 colony to the molar Hinton agar to confirm this result. All the bacteria gave minimum inhibition concentration in the second quarter, 100 milligram. Except the Enterococcus faecalis, the MIC and the MBC, 200 milligram per ml.
It must be from colony counting now, how you are making simply with streaking, then you are concluding.
Coloniform hygiene.
Yeah, CFC, counting must be there now. In order to conclude that MIC and MBC, you must do portrait technique.
Yeah, by separating technique.
No. Just from this well, you have to take 0.1, then you have to spread it, then you have to count the less than 2 days to count, to 2 more to count, 300. That you must do. It may not be a valid proof. And my concern is that MIC and MBC is quite high. Yeah. It must be with low concentration. Otherwise, it may not be considered for paper. That you must focus on. Okay. Anybody else?
I have a small suggestion that doing this MIC concentration, we have to keep a extract blank. Extract is having a colored extract, so you should keep an extract blank for the same. Otherwise, you can't compare the control samples. Usually, the positive control should be in.
I have control positive and control negative. This well control negative. This well control positive.
Negative control should be in purple color and the positive control should be in pink color. And the extract control should be in extract colored format. So it is a suggestion you can take up further studies. Thank you.
Okay.
Next presentation.
Thank you.
So namaste, good afternoon everyone. My name is Sijo. I am currently pursuing PhD in microbiology at the School of Bioscience, McFast College under the guidance of Dr. Smita Vijayan. So today I am presenting my part of my PhD topic, that is drug-resistant Klebsiella pneumoniae in clinical settings, implications for infection prevention and control. So Klebsiella pneumoniae is a gram-negative, non-motile encapsulated bacterium, and it is a major cause of hospital-acquired infections like pneumonia, urinary tract infections, sepsis and wound infections, and it can affect immunocompromised and critically ill patients. So the emergence of multi-drug resistant and carbapenem resistant strains causing a major global crisis, and the pathogen is recognized as by WHO and CDC as a top priority pathogen for AMR research. So they are resistant to beta-lactam, fluoroquinolones and colistin limits treatment options. So the AMR in clinical settings can spill over into environment and animals. Inappropriate antibiotic use and poor infection control accelerate resistance across systems. So there is an urgent need call for One Health-based surveillance and stewardship. So the study objectives includes isolation and identification of Klebsiella pneumoniae strains from clinical samples to evaluate the susceptibility of Klebsiella pneumoniae to commonly used antibiotics in clinical settings, and phenotypic characterization of AMR of the clinical isolates. So the study conducted in the Clinical Microbiology Laboratory of Pushpagiri Medical College Hospital during the period from January to March 2024, and it's a prospective cross-sectional study, and the study was conducted after obtaining the ethical clearance from Pushpagiri Research Center, and the study conducted with the help of under the guidance of Dr. Tina Jacob, Clinical Microbiologist at the Department of Microbiology, Pushpagiri Medical College Hospital. The clinical sample size was calculated using the statistical formula n is equal to z square pq by d square. Hence, the study is conducted in a minimum of 206 Klebsiella isolates. So, the study population included patients of all ages and genders, and included those visiting both outpatient and inpatient department of the hospital. Exclusion criteria are bacterial isolates other than Klebsiella, repeat isolates from the same species, and isolates obtained from stool samples. So the study began with the collection of clinical data, which involves age and gender, and the different specimens like urine, sputum, pus and wound samples were collected. The blood were collected and it is incubated in Bacti alert blood culture system. So and the clinical isolates are inoculated on both different medias like Mekong agar and blood agar. After incubation, the bacteria were identified on the basis of colony morphology, gram staining and biochemical test. And the antimicrobial sensitivity determined through Kirby-Bohr disk diffusion test performed on Muller-Hindon agar, and the phenotypic characterization of AMR confirmed through ESBL, AMC and MSIM and ESIM test. So the ESBL enzyme production confirmed by disk diffusion test, AMC enzyme production by cephositylene closacillin double disk synergy test, and carbapenemase enzyme production confirmed through MSIM and ESIM test. And colistin is a drug that is used for treating severe infections, and it is a last resort drug, and its concentration is determined by colistin broth dissolution method. And all these tests are done as per CLSI M100 2023 guidelines. Research. So on blood agar, they are large mucoid gray white non-hemolytic colonies. On Mekong agar, they are large mucoid lactose fermenting colonies. And gram staining, they are gram-negative state rods with the rounded ends arranged in singly or in pairs. And total 8 biochemical test results showed positive results for Klebsiella pneumonia. And sample-wise distribution, a total of 222 isolates were included after screening, and most of the isolates were obtained from urine, that is 150, followed by pus, blood and other fluids, and most of the isolates are obtained from the age group 61 to 75 years, followed by 46 to 60, and least in 0 to 15 years. And most of the infections were more common in males than females, males 123 and females were 99 cases. So the maximum antibiotic sensitivity, what we found that maximum sensitivity is found within tetracycline around 156, followed by ertapenem and imipenem. And Klebsiella pneumoniae is naturally resistant to ampicillin. And in Pushpagiri Hospital for severe infection and first empirical therapy treatment, we are using piperacillin tazobactam, and its resistance is around 39.1% and sensitivity is around 60.81%. and 141 were confirmed as ESBL producers, and maximum number of ESBL producers were isolated from urine followed by pus, and 120 Klebsiella pneumoniae isolates were confirmed as AMC beta-lactamase producers by AMC disc test, and 55 isolates were confirmed as carbapenemase producers by MSIM and eSIM test. So colistin MIC determined by CBDE method, and majority of the isolates had a colistin MIC less than 1 microgram per ml, and the urine samples had the highest number of isolates. First sample showed the highest resistance. Summary: a total of 222 Klebsiella pneumoniae bacteria were collected from different patient samples. These samples were tested for resistance to antibiotics using standard lab tests. About 66% of the bacteria were resistant to multiple antibiotics, and high resistance was seen in common antibiotics like beta-lactam, levofloxacin, trimethoprim, sulfamethoxazole. 39% of the samples were resistant to piperacillin tazobactam, a commonly used treatment that raises concern about the effectiveness of correct empirical therapies. Out of 67 samples tested for colistin, 4 were resistant. Resistance enzymes found is ESBL 64%, AMC 54%, and carbapenemase around 24%. Conclusion: drug-resistant Klebsiella pneumonia poses a critical challenge to infection prevention and control in hospital environment. Many common antibiotics are becoming less effective, so there is an urgent need for early detection, rational antibiotic use, and strengthened antimicrobial stewardship to combat the rising burden of AMR in clinical settings. Thank you for listening.
Any queries?
You made a wonderful presentation. Huh? How you made that flow charts and all, like cartoons? It was very impressive.
Sir, the icons, figures are created using BioRender. And also in National Institute of Health, United States, in their website, they have nowadays BioArt. So there are different icons are there. So for each subject, biochemistry, microbiology, anatomy, physiology. So on the basis of our topic, we can choose different icons.
Wonderful. Then using that formula, you found something 206 Klebsiella in total, but whereas you found 222 something. How this difference?
Sir, the clinical sample size determined with the help of epidemiologist in Pushpuri Medical College. So the sample size is around 206. But in our study, we got around 222, above 222. So my clinical co-investigator told me that we can take that, we can round that number. More than 206. Below 206, the sample size is invalid. So we can consider above 206. So we finalize the 222.
How did you choose different antibiotics?
Already all these antibiotics are on the basis of CLSA guidelines, sir, clinical laboratory standard guidelines. So if any patients admitting in the hospital, for all the samples we are testing all these antibiotics according to CLSA guidelines.
Thank you, sir.
Thank you.
Thank you. Now it's time for tea break. After 10 minutes, we'll join back here. Tea is arranged outside the hall.
Ada satu
ചളി കളിക്കാൻ പറഞ്ഞു സാരി.
എൻ്റെ ഫോണാണ് പറയും
Now, let's continue with the oral presentation.
Good afternoon. I'm Afafa Abdelkarim, research scholar at McFast College. I'm here to present my presentation, my topic, the role of nanocurcumin in protecting certain gut microbiota from malaria-induced toxicity under the guidance of Dr. Smith Vision. Gut microbiota is a complex microbial community in the gastrointestinal tract that is essential for maintaining human health. Malathion, an organophosphate insecticide, is widely used but can disrupt the gut microbiota and impair its essential functions. Nanocurcumin, a more bioavailable form of curcumin, supports gut health by reducing harmful bacteria, beneficial microbes, and providing anti-inflammatory and antioxidant effects. The aim and significance of this study and evaluate the nanocurcumin's potential to restore gut microbiota disrupted by malathion, highlight its antioxidant and microbial modulating benefits for gut health. Our research journey started by the extraction of curcumin from turmeric powder by the succinylate method at 60 degrees centigrade for 8 hours. Then concentrated and lyophilized to obtain dry curcumin. The second step was the identification of curcumin by using the high-performance liquid chromatography using chloroform and methanol solvents. After identification of curcumin, we used the preparative HPT-LC to isolate the curcumin in the silica gel. After that, we synthesized the nanocurcumin by the sonication method and study the characteristics of nanocurcumin, UV, VIS, FTIR, DLS, zeta potential, TIM, and XRD, and study the antioxidant activity of nanocurcumin. And finally, we study the effect of malathion using concentration between 0.2 to 3.2 milligram per ml, and study the effect of nanocurcumin 0.2 to 1 milligram per ml on selected gut microbiota. And then we study the restorative effect of nanocurcumin against malathion using the optimal concentration 0.8 milligram. I used 2TC for this steps and also used the colony-forming unit also to study the effect of malathion in selected gut microbiota and the restorative role of nanocurcumin. The results, the extraction, this picture shows the succinylate extraction of curcumin from turmeric by the succinylate method. Then picture number 2 shows the identification of curcumin by using the HPT-LC by comparing the results with the standard. And the third picture shows the isolation of the curcumin by using the preparative HPT-LC. Synthesis of nanocurcumin, I follow the following steps. Dissolve the curcumin in ethanol, then we add the curcumin solution to the boiling water gradually under sonication, and then stirring for 13 minutes, drying and storage. The results, after that we study the characteristics of nanocurcumin to confirm the synthesis of nanocurcumin. We use the UV-VIS spectroscopy analysis. UV-VIS analysis showed that the curcumin and nanocurcumin had absorption peak the same, 423 nanometer, but the nanocurcumin show higher absorbance. For nanocurcumin indicate the improved dispersibility and stability. For FTIR spectral analysis, also the curcumin and nanocurcumin showed the same functional group, but shift in the OH stretching band from 3,500 to 3,300 in nanocurcumin, suggested altered hydrogen bonding due to encapsulation. For DLS and zeta potential, DLS analysis showed nanocurcumin had uniform size of 131 nanometer, indicating monodispersity and successful nanoparticle formation, and zeta potential was minus 75.8, also confirmed the synthesis of nanocurcumin. For TIM analysis, the nanocurcumin showed spherical particles ranging from 21 to 36 nanometer, averaging 29 nanometer. We measure the size by the MHG in the first picture, and we draw the histogram by the origin. XRD analysis of curcumin and nanocurcumin. XRD X-ray diffraction showed that pure curcumin is highly crystalline with sharp peaks and crystalline size of approximately 34 nanometer. By Cher equation, we used Cher equation to determine the crystalline size, while nanocurcumin exhibited broader peaks and reduced crystallinity with a smaller size of approximately 27 nanometer. This indicates that the curcumin transformed to more amorphous, become more amorphous when transformed to nanocurcumin. This table shows the differences in the result between the nanocurcumin and curcumin in the TIM and XRD. We see the size is similar in the TIM result and XRD for the nanocurcumin. It is 29 for nanocurcumin by TIM and 27 nanometer in XRD. Antioxidant, the nanocurcumin show good antioxidant activity in the DBPH assay with IC15 inhibitory concentration 19.9 in comparison with the ascorbic acid as standard. Also show good antioxidant activity in FRAB assay with effective concentration 50.7 in comparison with the ascorbic acid. This is the picture show the TTC assay. In this test, we study the impact of malathion on viability of selected gut microbiota. Triphenyl tetrazolium chloride, this is colorless substances. But the viable bacteria, enzymes, especially dehydrogenase, can reduce these substances to formazan and produce the red color. So the red color indicates the viability and the metabolic activity. The disappearance of the color indicates the death and no metabolic activity of the bacteria. So we can notice with different species of lactobacillus and bacillus coagulans, when the malathion concentration increase, the metabolic activity and the viability of the bacteria decrease because the red color also decrease. Here we study the role of nanocurcumin in restoring the gut microbiota. We can notice, also we use the TTC to notice how the viability decreased when using the malathion and how the viability restored when adding the nanocurcumin. For each picture you can notice M, in the next picture M plus nanocurcumin. In the presence of malathion with selected gut microbiota only, the viability was low because the red color or formazan formation was low. In the next tube, we noticed that the color increased, the red color increased. That indicates the increased viability. That means the nanocurcumin enhanced the growth of selected gut microbiota and enhanced the metabolic activity of the selected gut microbiota. This is study the restorative effect of nanocurcumin by using the colony forming unit. We can see the control growth, the growth with malathion. You can notice how the colony forming units decrease and how the nanocurcumin restore the growth again. Here, this table shows the numbers of the colony forming units in the presence of malathion, in the presence of nanocurcumin, and control. Conclusion, nanocurcumin was successfully extracted, purified, and characterized, exhibited enhanced stability, dispersibility, and antioxidant activity. Malathion significantly reduced the viability and metabolic activity of selected gut microbiota. Co-treatment with nanocurcumin restored bacterial viability in concentration-dependent manner by using TTC assay and colony forming unit. Protective effects are likely mediated through antioxidant and microbiota modulatory properties of nanocurcumin. These findings highlight nanocurcumin's potential as therapeutic agent to counteract malathion-induced gut dysbiosis. Thank you.
Any queries?
Do you have query?
No.
So you have performed the statistical analysis, no? Statistical p-value, significant test.
We just here get the mean and standard deviation and p-value also, yes.
Yeah, so how did you do, what kind of significant tests did you carry out to find the p-values?
P-value here indicate that, yes, the differences in the color has significance. That means because we get significance in all the results All the p-value
No,
less
I'm
than...
just asking. There is ANOVA, there is different Kruskal values and different test of significance. So, in this, what kind of test of significant test did you perform for finding the p-value?
ANOVA, yes, ANOVA.
You used ANOVA?
One way or two way?
Yeah.
Duncan, Lamproni, likely there are many tests, just you specify what you are. Because one way, you have only one observation, you had only one thing, like there are some observation numbers to consider a statistical test. So overall, which ANOVA you all used for all data, did you use only one statistical test, that is one ANOVA or some other thing?
Yeah, we just use ANOVA after we just get the mean, standard deviation, and ANOVA to get the significance level.
So there is another question. So how did you decide that ANOVA is the perfect test here?
I think according to reference also, I noticed that.
That is fine. I mean, generally based on the distribution of the data, you choose which kind of test of significance. If it is normal distribution, then you have to probably go for ANOVA. If it is non-normal distribution, there are other tests. So I just wanted to know. It was wonderful work. I really loved it, but I just wanted to know this part.
He's asking a valid question. Actually, you should focus this also, because mostly it was showing a highly significant value. Only the last case it is showing a moderately significant. It is less than 0.05. So you must consider. Then one more thing, go back. How I want to know the difference. What makes that sonication into nanocurcumin and curcumin?
Why I used nanocurcumin? Because
no, what makes the difference between a curcumin and nanocurcumin? There you didn't use any precursor or substrate. Only you went with curcumin. So what made that nano valuable here?
Okay. The problem of the curcumin, it is low soluble. So it has absorbance in the body is very low. It is approximately 1% only. So we go to nanocurcumin to enhance the absorption and bioavailability in the body.
How you made that nanocurcumin? Just I want the mechanism.
By sonication method. By sonication method. We use
sonication. What is happening?
Yeah, we dissolve the curcumin in ethanol. After we get the solution, we drop the solution gradually to the distilled water during sonication. After that, after adding the solution to the distilled water, we do stirring for 30 minutes and then centrifuge, washing the particles, and then drying the sediment.
So that nano comes only to reduce the structure of curcumin to nano size, or because you had a 423 nanometer. You know, each metal or metal nanoparticle has a certain range. Now yours is 423. So it is because of silver, I think. 438 something around. So how you confirming with UV that 420 nanometer is nanocurcumin confirmation initial characterization?
We used TIM. The size was?
No, that range for metals when extract or any plant source or any reagent being, that is substrate. Precursor is silver nitrate, like when you add biological synthesis, then we may get some nanoparticles. So that had a range when we can confirm from 420 to 440, like the 427 comes means then it's a silver. When it is 545 and 555 it is gold. Likely we have certain ranges for different metals, but in your case you just mentioned nanocurcumin without any precursor. So how you are confirming that 428 is a nano?
We compare between the curcumin and nanocurcumin in many tests. At first we compare between them in the UV. The UV absorbance difference become differences indicate the increased absorption in the UV, because the absorption of nanocurcumin was very high in comparison with the curcumin. That's indicate the high absorbance, high solubility and disapparity. Also, there is comparison in the functional group. The OH, there is shift in the nanocurcumin that indicate the change in the OH group.
When structurally some modification is happening, then it won't be a curcumin. Then how that shift all and all is saying as nano? What confers here as nano? That is my question.
I confirmed that by the comparison between curcumin and nanocurcumin, there is change in the UV, FTIR, And also I confirm by the TIM result and XRD, the crystallinity decreased between curcumin and nanocurcumin. This is the result of the XRD. For curcumin, the crystalline size 34 nanometer by Chair equation. But for the nanocurcumin, approximately 27 nanocurcumin. That means it becomes more amorphous. That indicates it becomes smaller size and more soluble.
And another thing, you have mentioned zeta potential as minus 73. It's not a stable one, you know. Did you notice that? Minus
For,
73.
zeta.
It must be minus 10 to minus 30 plus or minus 30. You know, that range must be, then only the nano will be confirmed.
I know. For the less.
Here.
Yeah,
See, minus 75 is not a valid result.
Yeah.
Did you publish this data or still under
Okay,
consideration?
minus 7.5, it is between minus 13 to plus 13. It is between this range. This is the range.
That was my question. It's not a valid result.
What is the exact value?
For zeta.
For zeta, it is minus 75, minus 7.5. What is the exact value? Minus 75.
See,
It is minus 75.
it's not a good result for confirmation of stability, charge-based stability with 75. You can refer in Google.
What is the point is that when you are showing a nano
Minus 10.
size, especially in the range of 75, minus 75 zeta potential, okay?
Yeah.
That means that your particle is highly unstable. Okay, the standard range is minus 3 to minus 30 like that. All,
It is
or
the stable
minus
range.
10 to 30, minus 30. Okay, that is a standard size. Beyond that, if any particle is forming like that, because it's beyond minus 30 means, or less than minus 10 means, the particle is not stable in the solution. Okay, so when the particle is not stable in the solution, so your particle will, agglomeration will come. It will settle down in the bottom. It will be not in a uniform distribution.